Good afternoon, class 12 children. Today we will again continue with chapter 14 in bio of biotechnology and its applications. We saw in video 8, we studied about the importance of DNA fingerprinting. We saw the forensic DNA fingerprinting and how it is utilized to catch culprits, etc. We learned about the hybridoma technology, where fusion of cells is involved and indefinite multiplication of hybrid cells takes place. We studied about very important molecular diagnosis for effective treatment of diseases and its diagnosis and understanding of the pathogenicity of the disease. We studied about three molecular diagnostic techniques, namely recombinant DNA technology, polymerase chain reaction or PCR, and about enzyme linked immunosorbent assay technique or ELISA technique. Today, let us see what is ELISA technology based on. It's based on two principles upon antibodies and some antigens can attach to polystyrene plastic plates or solid phase supports and still maintain their full immunological capabilities. Now let me explain what is written here, children. We have got plastic bowls or plastic trays or plastic plates. Now these are made of polystyrene plastic material. So in this we carry on the ELISA test, and in these plates the solid phase supports and still it maintains full immunological capabilities. We will see and understand this further through an experiment. Then we see that antigens and these antigens can be bounded to enzymes. The antigens are those organism or those particles which cause infection in a person, and these antigens, they can be bounded to enzymes, and the resulting complex are still fully functional both immunologically and enzymatically. We see, if you remember, in class 11th we had studied, children, about the enzyme substrate complex. So here the enzyme, it takes on the antigens that fix to the active site on of the enzymes, and it makes a complex. It is the enzyme activity which is the measure of quantity of antigen or antibody present in the test sample. So we can find out, even if the quantity we know yesterday it is present 10 to the power minus 10. So this much less quantity also can be detected by ELISA test, or if more of the quantity of antigen or antibody is there, then also the test sample can be studied and the antibodies or antigen presence can be detected. Now the two methods of enzyme immunoassay that have been found to be of most clinical value are the double antibody sandwich procedure for detection and measurement of antigen, and the other procedure is known as the indirect microplate ELISA procedure for detection and measurement of antibodies. Keep looking at this diagram, children, as we explain these one by one. Now pay attention here. The first method of ELISA technology, how it is used. Now, this children is the rough diagram shown of the microplate that I was talking about, the polypyrene kind of plate. So this is the plate that I was talking about. So this step involves the coating of polystyrene wells. This is a polystyrene well you see here. In this polystyrene well, we see that the antigen is passively absorbed. Do you see the antigen here? This antigen is passively absorbed, or it sticks at the very base of this trough, which is made of polystyrene material. Here the antibody is absorbed to the well surface. Now the second step here, children, listen carefully, only then you can understand, children, the second step. The antibodies in the anti-serum adhere to the surface of each well. Do you see this antibody in the anti-serum? This antibody adheres to the surface of each well, and the test antigen is added like you see here in this figure 6a, that the test antigen is added, and this antigen binds to the antibody which was already sticking here. And if it is homologous, that means it has the right kind of site, then this particular test antigen would find its site on this antibody and stick into it. Then we see that the third step, then the test antigen is added. You see here, the test antigen here is added, and if the antigen is homologous, it sticks to the antibody immobilized on the well surface. So this antibody is immobilized, this cannot move, and onto it the test antigen has come and has stuck onto it. Then we see here that enzyme labeled specific antibody is then added. This is a specific antibody, children. So, and this is enzyme labeled. Can you see here? 1, 2, 3. The enzymes are labeled here, and this well binds to the antigen already fixed onto the antibody. So this results in an antibody with enzyme. Here you see this is an antibody with enzyme, and antigen and antibody sandwich. Can you understand this now, children? Again, let me explain. Here this is an enzymatic antibody which has an antigen after it, and again an antibody here. So it is an enzymatic antibody, antigen and antibody complex. The rate of enzyme action is directly proportional to the quantity of the enzymes labeled antibodies. As much as the enzyme labeled antibodies are, so much would be the rate of enzyme action. In turn is proportional to the amount of test antigen. Now this also the reaction of the antibodies with the enzymes also depends upon the test antigens, how much test antigens are found. The enzyme activity may be followed by a color change. So you see here that this is the enzymatic activity that is happening. You see the active sites of these enzymes, they are able to stick on the various particles here, and these particles or the substrate, it's written S, all these substrates, they try and find the active sites, and this can be inspected visually or measured by the means of colorimeter. Colorimeter is a meter which sees how much color is there, and this method is used to assay. Assay here means test. This method is used to assay or test the hepatitis B antigen. The other method that we see here, children, is known as the indirect method, or this is also, children, known as the indirect microplate ELISA procedure. The initial step involves the coating of polystyrene wells with antigen. Now here we started on with the antibody, but here in this kind of test, which is indirect microplate ELISA procedure, in this first the antigen comes and immobilizes at the bottom of the polypyrene well here, and once the antigen is absorbed passively to the wall of this well, then we see the test antiserum. Do you see here? The test antiserum is added and is allowed to incubate. This test antiserum is added and it's allowed to incubate. Incubation time is the time it takes so as to bind to the antigen that is provided here. So this antibody, it binds to the antigen as it comes out of the antiserum. If the antibodies in the antiserum are homogeneous, if these antibodies are homogeneous, they will bind to the immobilized antigen. Do you see here? It has got the right kind of site, this antigen, so as to accommodate this antibody, and so this antibody comes and fixes itself over this antigen. Then we see that the enzyme labeled anti-human antibodies are added. These are the enzyme labeled anti-human antibodies, which you can see in the third step. These are added to the system, which will link to the antibody. So this was the antibody, and this particular anti-human system comes, enzymatic system comes and attaches itself to the antibody antigen complex. This is the antibody antigen complex, so it comes and it fixes itself on top of it. Finally, the enzyme substrate is added, and the rate of its degradation, the substrate used up, or the degradation of the substrate, is associated with the color change proportional to the concentration of the antibody present in the test sample. So the coloration will tell that how much of antibody is present in the test sample, and this color change can be monitored visually by our eyes, or also colorimeter, which tests the amount of color is used. This method is used for the immunodiagnosis of many infectious pathogens. So this is how we use the ELISA method. Coming to another method that is used is biomedical engineering. With the advancement in medical technology, number of instruments and devices have been developed that have superbly contributed to our medical world. Biomedical engineering provides sophisticated equipments in modern hospitals for diagnosis and treatment of patients, like ECG, electrocardiography. It functions, how it functions, the ECG or the electrocardiography, it records the electric activity of the heart during cardiac cycle. that whether the electric activity of the heart is normal or there is some variation in that. Then we see electron cephalography or the EEG. This records the electric activity of the brain. We see another biomedical engineering method as EMG or electromyography. This records the muscle action potential or the muscle tone. Then we see another biomedical engineering technique which is called as auto analyzers. The auto analyzers, they are fully automatic computer controlled devices, and these devices are used to estimate various biochemical substances in body fluids. How many biochemical substances are available in the various body fluids. Then we also use X-rays, which are imaging instruments when it began. So we do X-rays. Then came more sophisticated imaging instruments like tomographic scanning instruments. Then we see also positron emission tomographic scanning or the PET scanning instrument. Then we see NMR or the nuclear magnetic resonance imaging. All these are imaging which takes the images, or they can sketch the images from inside of our body, inside of our organs. Then ultrasound imaging or sonography is very well known, which is done for liver, for stomach, for spleen, and so many other organs. Then we see therapeutic instruments are used for curing diseases. The therapeutic instruments are pacemakers, medical pacemakers, intra-aortic pumps or pumps which restore functioning of the heart. They are used. Then we see implants that are used. Some of the implants have been created by Indian scientists, like artificial heart valves were created, and vascular grafts in the form of artificial arteries, they are implanted in humans when the natural arteries fail, and joint implant is also used. Then we also see blood dialysis or the blood dialyser, which is also known as the artificial kidney. This clears the blood from these poisonous waste products and makes the blood clean. Then we also see there are other biotechnological instruments called biosensors. For what are they used for? They are used for measurements and for recalibration or the counting procedures, and are low cost devices for repeated measurements. These are disposable devices which can be used and thrown, because so many patients, they visit the hospitals on a daily basis. Then we see minimal sample preparation is needed and continuous measurements are usually obtainable through the biosensors. Then biosensors, there is a demand for devices to measure specific biological metabolites. Example, the metabolites present in a body are glucose, cholesterol, metal ions, and hormones. Then we see biosensors are used not only in medicine but also in agriculture, in the field of agriculture, for monitoring or for finding biological activity in the livestock. What is livestock? Like cattle, buffaloes, goats, cows, in the livestock, and detection of viral and pathogenic infections in the cattle, in cows, etc. And quality control of dairy products. There also biosensors are utilized. So children, we will stop the video here and we will follow in the next video. So wish you all the best. Have a good day. Thank you.